There is growing appreciation for the impact of the mechanical microenvironment on cell signaling and migration, necessitating the use of unique experimental approaches to deliver mechanical stimulation to individual cells. This technique allows the dynamic manipulation of the mechanical microenvironment underneath a cell in real time, allowing the observation of mechanically regulated changes in cell migration behavior and subcellular signaling events. Demonstrating the procedure with me will be Nyla Naim, a postdoc, and Johnathan Patterson, a technician from our laboratory.
Begin by using a corona wand to activate the glass surface of each bottom cover slip for 20 seconds, before immediately overlaying 50 microliters of bind silane working solution onto each cover slip. Allow the solution to dry for 10 minutes, before rinsing the cover slips 2 times with 95% ethanol and 2 times with isopropanol. Then, allow the cover slips to air dry for approximately 20 minutes. For fluorescent microbead deposition, first, sonicate the working microbead solution for 1 hour. 15 minutes before the end of the sonication, place the activated cover slips vertically in a ceramic cover slip holder, and transfer the holder into a tabletop plasma cleaner for room air plasma treatment for 3 minutes. Next, place pieces of paraffin film in 60mm Petri dish lids, and place the cover slips onto the films, lightly tapping each cover slip to ensure a good contact between the film and the cover slip. Then, add 150 microliters of the working bead solution to the top of each cover slip, and